I received some criticism from ladies that the gender balance at this meeting is not proper. 14 males against one fragile lady. But now you will see something interesting. The gender balance is okay. Because if you hear Mara, you will realize immediately 14 males, macho males, cannot balance her out. She's so fantastic, so dynamic, and so intriguing because she's a medical doctor who is in science, a scientist who's in medicine, so she's traveling between the two worlds. But two is not enough. Yesterday we heard from somebody about the triunite brain. So she has a third brain. She's a professional musician, and with her husband and 4 grown-up sons, they have a band. So next time she promised me to give a concert. So we very much look forward, Mara, to your next visit in Singapore, when you will hopefully have your band with you, I mean sons and husband also, and give a concert. But now we would like to listen to your lecture, which I promise will be a balancing act.
Okay, thank you. Thank you, Maria. So, can I switch to my presentation? Thank you. So first of all, I'm very pleased to be here. It's really great. It's my first time here in Singapore. And I'm very pleased to have been in this gorgeous conference. My talk, and now comes a disclaimer, is completely different. So my disclaimer is I will not speak about oscillations. Sorry about that. Instead, I will talk about our work on trying to find therapies for brain disorders. And this is in part because of my medical background. So I'm interested in understanding, but I'm also interested in applying this understanding to brain disorders, in this case, to society. So why do I call it we need a shift in paradigm? I will try to explain it very kind of superficial if you want. We can discuss later on. But first, let's think of what is brain disorders and what is mental health. And this sentence is not mine. There is no health without mental health. This comes from these Lancet papers in which the authors were claiming that mental health is not just the absence of mental disorder, but is what helps us supporting well-being, good life, and also productivity, right? So the problem is that, in fact, in the last years, due to different reasons, the economic crisis, the conflicts around the world, the way we are organizing our society is increasing. And somebody was saying, and somebody was saying about the increase in self-harm in women in UK. And most of these disorders are influenced, greatly influenced, by these societal changes. So right now, and I take the numbers in Europe, that is where I come from, in fact, brain disorders will affect at least 1 in 3 people. And I am including not only, let's say, neurological disorders, but of course, mental disorders, intellectual disability, etc. Okay? The cost exceeds far the cost of cardiovascular disorders, cancer, and diabetes combined. Okay? It's a very big problem. And this accounts for around 35% of the burden of all diseases in Europe. So the curious thing is that despite this very urgent need, clinical need, we have made very little progress in the last year in psychopharmacology, right? So in fact, most of our prescribed drugs are based on decades-old science, right? And this is reflected in the numbers. There are very few new products that really go far into the clinical practice. And these affect especially things like neurology or, in general, mental health, mental disorders. It is not because we know less. We know far more. And this is just an example. You have heard excellent examples of the amount of knowledge we are getting about how the brain works, but also about the genetic cause of different disorders. And this is just part of the number of genes that have been associated to different mental disorders, right? So the problem is that these advances in the understanding of the molecular basis of disease are not translated into real therapeutic targets, right? And in fact, this is a paper a couple of years ago about what the physicians think are the most transformative drugs of the past 25 years in all fields, right? And in fact, 2% of the assessments really reach this level of transformative therapies. And what happens is that most of the new compounds are only marginally better, but much more expensive, of course, because the business model has to go on, you know. Okay, so what is the consequence of that? The consequence is that most of the large pharmaceutical companies have removed the psychiatric diseases from, let's say, its body of discovery, okay? So, and in fact, this is taken from, I like very much this metaphor that Stephen Hyman in his paper in 2012 was taking to explain the situation of these big pharmas. And if you look at the causes, if you try to understand, so what are we missing? First, there is still a lack of understanding. We don't know how all these genes that we are finding associated to disorders, to disease, are really acting to produce these disorders. We don't know that. We don't know how is the natural history. We take a snapshot. We take a snapshot when everything has already probably happened. We take this patient, oh, he has symptoms. Let's cure the symptoms, right? So this is also a problem. We also lack good disease models, and I will briefly explain why. We also lack good biomarkers. And we mainly lack translational research. Okay? And in fact, we still deal with this concept of the magic bullet, right? That comes from old, let's say, discoveries like the one made by John Joseph Cade in the 40s that found that lithium had sedative properties. And after that, there were a lot of path-breaking discoveries that really changed the whole field of psychopharmacology, and it was amazing for the psychiatric patients. But the problem is that those do not cure the disorders. They treat the symptoms. And they are mainly neurotransmitter-based therapies, so chemical therapies, trying to restore the chemical balance in the brain. So, in fact, when we look at the main drug classes that we use, they are still coming from this tradition, right? And also, if we look at what is going on now, they are being used for other things, right? But with the same type of concept. There are some new drugs that are different, but there are very few. And what happens now is that we have more and more medicalization also. So we are also treating people that not every time should be treated. And this is something that, as a society, we should also take into account. Besides, we have relatively bad models for at least some of these psychiatric disorders. So the depression, for example, is based on inflicting stress to the animals in different ways. And then we do the transcriptomics, and we are not sure what are we understanding, if stress-related disorders or depression or what exactly, no? So this is also a problem that we have. So we need a shift in paradigm. And now I could finish my talk, no? Because I already convinced you. But I will show you some example of how we have, in my lab, done this, try to do this shift. So the idea is to go from the symptomatic treatments, mainly neurotransmitter-based, to what we could call disease-modifying treatments based on understanding better the pathophysiology, but also prevention. So if we can really identify good biomarkers that tell us when something starts to happen, probably we will be more able to prevent the appearance of these disorders, right? This could also be based on targets from genetics, very clearly, but we need to understand better what is going on. And what I will focus on in my talk is a very specific thing, that is a mechanism that may promote synaptic plasticity. And in fact, the idea is to go from this concept of magic bullets, that is these maximally selective molecules, right, for specific targets. And I have to also refer to the talk yesterday about the Western and Eastern differences. There is another layer of difference, that is the fact, and sorry guys, that science has been dominated for hundreds of years by a reduced number of males, not exactly representative of all the population, right? So I think that this concept of the magic bullet, that I will kill the receptor, right? I was saying this to somebody the other day. And the more potent I get it, the better, right? So maybe we should go to something probably less astonishing in first place, but more effective in the very end. That is what is now called the network pharmacology. So here, instead of basing the concept in molecules, we base our treatment in process. Try to restore the process, not the molecule, right? Okay, so let me drive you through our particular journey through translational research. And in my lab, we are interested mainly in cognition, so intellectual disability is this problem, this generalized problem that interferes with many cognitive domains. And specifically, we have worked in several disorders, but one of our main contributions have been done in the field of Down syndrome. So Down syndrome is caused by the presence of 3 copies of the chromosome 21. What does this mean? At the end of the day, what this means is that we have a bunch of genes, of normal genes that are not mutated, that are encoding for perfectly normal proteins, but that are creating a disbalance, an overdose of many of these proteins, okay? And this overdosage is what is producing the phenotype that we know. So basically what we know is that they have cognitive deficits. They have many other problems in other organs and tissues, but I will concentrate on the brain phenotypes, right? So basically they have problems in cognition. They have different memory and learning problems, language problems, executive function problems, and they have also underlying alterations in different brain regions. So some of them are macroscopic, so you can see that the brain is rounder, and this is an interesting issue that if you want we can discuss at the very end. But also, so they are brachycephalic, and they have, let's say, reduced volume that is relatively specific of some brain regions. Some of them have been highlighted here during these days, the cerebral cortex, the hippocampus, but also another region that I think is very important, that is the cerebellum. Today I will concentrate on the cortico-hippocampal system, right? And what we see is that besides these macroscopic alterations, we have also alterations at the microscopic level. And in fact, if you would think of a neuron as a cell able to communicate with other cells, as has been highlighted by my colleagues, if we look at the communication system that is composed by these membrane specialization called dendrites that receive the information, and these little dots here that are the dendritic spines that are the entrance point of excitatory information in the cell. And all these things help to then take cellular decision that will then propagate through a structure that is called the axon to the next neuron. So sorry, this is just for the non-neuroscientists so that you can follow me a little. Okay, so basically you can imagine that this may have a consequence in how the neuron is able to communicate and to process the information. And this is what we call the wiring problem in intellectual disability. And sorry for ID means intellectual disability, right? So in many different mental disorders and brain disorders and intellectual disability disorders, we see that those structures are altered. And you can see this across many different disorders, Alzheimer's disease or neurodegenerative disorders, but also Down syndrome, Fragile X, Rett syndrome, autistic spectrum disorders, and all of them have sometimes different connectivity problems. So why is this important? Imagine that you have a brain and you have seen how all these overlapping networks are communicating and are creating different types of information at different scales. What we have to think is that cognition and behavior are emergent properties. Okay, so of course things like the architecture of the elements that are part of these networks is important and is probably determining or at least contributing to differences in the neural physiology. So this structure, in fact, can be fine-tuned by a process called plasticity. And when we learn or when we are developing our brains, plasticity is a very important process that is contributing to changes, to subtle or strong changes in the architecture of these circuits. Oops, sorry. Okay. So what happens in intellectual disability, we could imagine that this genetic disturbance is leading to alterations in both the target space, so where the neuron can reach, but also in the number of connecting points, let's say, of points of entrance of information. And this certainly has consequences at different levels, okay, at the systems level. So how do we tackle the problem in intellectual disability and specifically in Down syndrome? What we have in Down syndrome are models that are resembling the genetic cause. So the trisomy of these specific regions in chromosome 21. And there are different trisomies. So we have a whole zoo of aneuploid models. So meaning that they have excess or defect of number of chromosomes, right? So we have almost the whole equivalence, the whole homologous regions of the chromosome 21 in different trisomies. We have also full trisomies. We have also partial trisomies, right? And this helps us dissect a little bit what could be the genetic cause of the specific symptoms or signs that we are looking at. And the chromosome 21 has its homology regions distributed on chromosome 16, 10, and 17 in the mouse. So I will concentrate today in one model that is called the TS65TN, that is a partial trisomic 16, but that contains the genes that have been in genotype-phenotype screenings shown to be the most important for giving rise to the symptoms of Down syndrome, right? And this is called the Down syndrome critical region. We can discuss also later because there is a lot of things about that. Okay, so this is great. We can understand the biology behind this trisomy, and we can see how this gives rise to different symptoms, and we can see if therapies are useful. That's fine. The problem is that we cannot dissect what are the specific genes that may have a more important contribution to a phenotype, right? So to understand this, what we can do is we can select candidate genes and in an otherwise disomic context, only put an excess of those of this gene of interest, okay? So this will tell us if our gene is sufficient to produce a phenotype. Okay, so of course, when you have a mouse model, you have to prove that it's a good model of the disorder you want to model, right? And this is formalized in different validity criteria. So one is the construct validity. That means that you're trying to put in the model exactly the same causes, the same etiological factors that you have in the disorder, if you know them. But then you have the face validity, and this means that you have to have similar symptoms that the patients. And then you have something that is the most important, this is the predictive validity. That is, that the treatments, if the causes are the same, if the underlying biological mechanisms are similar, then the treatments that work in humans should also work in mice, and vice versa. Okay? So then what we do is a full, let's say, diagnostic test battery, which includes a lot of different behavioral tests in the mice. And in this mouse model, what we see is impulsivity, hyperactivity, reduced habituation, also clumsiness, impaired equilibrium, that is also present in Down syndrome, altered procedural learning, impaired coordination, and in the cognitive domain, we have different alterations in different domains. Okay? So I will just, as an example, focus on a very classical test many people use, that is called the Morris-Water maze, because my friend Richard Morris devised this idea, right? So here what we have is a pool. So you put the mice in the pool, and I will show you a little video on that. And based on these extra maze cues, he has to find its way to the platform. He wants to escape from the platform. He's like the kids. They don't like the water, except in the swimming pool. And what happens is that in the beginning, before the mice have learned, they tend to cover all the distance in the platform, searching for the platform because they don't know where it is. And then they start learning, and at the end, they are very efficient, and they go almost directly to the platform. And this is, you can formalize this in many different measures. I will just stick to one or two. But if you want, we can discuss how we analyze all these things, because it's, of course, a multidimensional behavior, right? So this gives rise to a progressive reduction in the escape latency, or in the distance traveled, or in many other things. And finally, you get to what we call the asymptotic level of learning. So this is the learning curve. And what happens in our trisomic mice is that, in fact, in the beginning, they don't get it. They cannot learn. And then they get kind of inspired, and after more intensive training, they can reach even almost the same levels as the wild types. But you see here in the wild types, they have a beautiful learning curve across all these sessions. So I show you here a video of a wild type in here, up, video, and of a trisomic, down. And you see that the wild type, very quickly, he makes a look, and he finds the platform. While the trisomic is not only that he takes more time, it's also the strategy that these mice are using that is very inefficient. They tend to repeat behaviors like this, that they know that they don't work, because they have been doing this for days, right? Okay, so besides that, what is behind? Do they also show similar alterations in the neuronal structure as the Down syndrome patients? Well, in fact, yes. And these are studies that we did. This was in the cerebral cortex. And these are horizontal cuts. So you cut the brains like this. And you inject this fluorescent dye that is called Lucifer yellow. Very nice name. And then you counter stain. So here you see the reconstruction of these control, the wild type. We call them wild type. So the normal mice, the euploid mice, are called wild type. So the control mice, and you see that the trisomics have smaller dendritic trees that are less complex, and on top of that, they have less dendritic spine. This is good news, because it means that the trisomy in mice gives rise to, and I have simplified a lot, of course, but more or less the same phenotypes as in humans. So this is good news.
What cells will be? What cells?
These are pyramidal cells layer 2-3 in the motor area. And this is, so the next thing was, okay, let's try to narrow down the genetic dependence of these effects. So how can we select among all these more than 300 genes, which are the ones that are more important? Because not all of them have the same effect. Some of them have really a dosage-dependent effect, but some of them may have no effect, so they are compensated, or there is epistatic compensation, or they can transact on other chromosomes. So you may have also alterations in the dosage or in the levels of expression of genes in other chromosomes, not just the chromosome 21, right? Okay, so we were testing, let's say, many different candidates, and some of them were pretty interesting candidates on paper, but then when you put them in overexpression alone, they give rise to no phenotype. But there was this one that is called DIAQ1A, dual specificity tyrosine phosphorylation regulated kinase 1. This was the name that the biochemist gave to this gene. The Drosophila people that are kind of more interesting people to some extent, they call it mini brain, because when mutated or when overexpressed, this change in the expression levels of DIAQ1A give rise to a smaller brain in Drosophila. So it's pretty well conserved, because in humans it gives rise to the same. So the deletions in the DIAQ1A region also give rise to microcephaly. So why is this gene interesting? Well, basically because it's an enzyme, it's a kinase. So it has regulatory properties. The kinases in biology are like interrupters. No, how is this called? Switches. Thank you. Sorry. And you can switch on or off many molecular cascades that are involved in different cell processes. And what is interesting of DIAQ1A is that many of these targets, phosphorylation targets, are related with very interesting properties like proliferation, so the creation of new neurons or adult neurogenesis also. It's also involved in Alzheimer's disease related protein phosphorylation like tau or APP. And it's very much involved with different cascades related to synaptic plasticity. So, what is the rationale now? What we can do is, as I explained, we can overexpress this gene alone, only overexpress this gene to see if it's sufficient. And then if we see that it creates the phenotype, then what we can do is we can delete it from the trisomic background and see if we can reverse the phenotype. Okay, so this is what we did. And this is just to show you that the levels of overexpression of this transgene are more or less in the range of what has been shown for the trisomic mice, but also for Down syndrome. This is postmortem fetal material, fetal brains. And the expression pattern is very similar to the one that we see in trisomy 21. So it has not ectopic expression that might happen. And what is interesting is that, in fact, just by overexpressing DIAQ1A in the same region, so the same type of neurons, we also get this phenotype, this reduced arborization and reduced complexity of the dendritic tree. And this is accompanied also by alterations in the learning process. So that was good news, because it seems that the overexpression of DIAQ1A was able to recapitulate at least these two phenotypes, right? The neuronal phenotype and the learning phenotype. So the next thing was, okay, let's try to see if we can reverse the phenotype in the trisomic mice. And in fact, normalizing the expression levels using sRNA constructions, so gene therapy, let's call it like this, in the brains of DIAQ1A, and we tag it with a fluorescent protein so that we can follow the expression levels of the SH-DIAQ1A with bioluminescence. And here you see that the distance travel, this is pre-injection, you see that they take more distance to find the platform, and then 2 more months after the treatment, the ones injected with the SH-DIAQ1A construction have similar levels as the wild type, while the scrambled construction, that is simply a control, or the saline injected mice have the RO version of the phenotype. And this was reproduced also in trisomic mice. So this is great, but the problem with that is that you cannot use it in patients. And as I was explaining in the beginning, one of the ideas was to try to then go back to the human. So fortunately, we found that some colleagues of us identified a molecule that is present in the green tea, that is a polyphenol of the green tea, is the most abundant catechin in green tea, that is called epigallocatechin 3-gallate. And this substance can cross the blood-brain barrier and has properties of deoxygenase kinase inhibitor, and at a very kind of convenient nanomolar inhibitory concentration. So we use this in transgenic mice to see if by normalizing the kinase activity of deoxygenase, not the expression of the gene, but the activity, okay, we were able to also rescue the learning phenotypes, and you see that the transgenic treated with this substance is performing the task as good or even better than the wild types. Here, the advantage is that you can go to the hippocampus, and this is because this task is related to visuospatial memory that has a certain dependence of the hippocampal integrity. And here you see that, in fact, the increased kinase activity of deoxygenase that we here measure biochemically in hippocampal extracts is normalized by the treatment. Okay? So this was good news. And so we decided to look at the hippocampal structure also. And first of all, we need to see how is the hippocampus. And to this aim, we did another genetic trick that was to cross our transgenic mouse model with a model that was expressing a fluorescent protein, the yellow fluorescent protein, driven by a promoter that directs the expression of the gene to the pyramidal neurons. And we create this double transgenic, and this is loved by my PhD students because they don't have to stain anything. They come already stained, so this is great for them. They love it. And here you see, this is the pyramidal layer in CA1, and you see the stratum radiatum. And here you see the same region in the transgenic mice. And you see that the whole system is pretty disorganized, and the dendrites are shorter. And they tend to have these kind of weird paths. So what happens after 1 month of treatment of epigallocatechin gallate orally administered? Well, what happens is that we rescue the phenotype. So this was, again, good news. But of course, as you know, and has been also highlighted, there is not just the genetic background. And this is something that is very important in Down syndrome. So in fact, the final phenotype that you can see is determined by the interaction between these genetics and the environment, right? And this is especially true, I mean, even though you have stereotyped structures at the macroscopic level, probably these environmental changes lead to also changes, as was highlighted before in Paul's talk, no? And of course, this gives rise to clear differences, no? Even though they have probably the same macroscopic structure in the hippocampus or in the cortex, they are very different, no? People. Okay, so, and this leads me to the term that I was mentioning before, the plasticity, the term plasticity. So it's not a perfect term, but let's take it as the capability of producing changes in the structure and the function of the brain at different levels. Of course, during development, the changes are more kind of severe, and in the adult, they are confined to specific regions and processes. But of course, this is something that is going on during the whole life of the individual, and it's contributing to brain development, and then in the adult, it's contributing to different processes we can also have maladaptive plasticity. That's also true. So, but, well, I cannot enter in all this. My point is that, in fact, one of the things that has been used during many, many, many years, not only in Down syndrome, but in general in intellectual disability, is this early intervention. And early intervention, but also the integration in the society of these people, have led during the last 40 years to increases in their IQ that are really significant. Okay? So it's really making an effect. The problem is that it's not as good as in the normal population, first, and also the effects tend to be temporary. Okay? So probably there is some alteration at the molecular level that is justifying these temporary effects. So we studied this in mice. Of course, it's not exactly the same model, right? But we can use what we call environmental enrichment. That is a way of inducing this activity-dependent plasticity, and this is one of the settings. This is from a paper in Nature Neuroscience. And I started these experiments with Jesus Flores. Jesus was my supervisor in my PhD, so I also wanted to highlight my supervisor. And he has 2 children with intellectual disability. But when I met him, he was studying opioids. So I started, my thesis was on central mechanisms of opioid action on the respiratory system, you can imagine. And I was always curious about why Jesus was not devoting some effort to try to understand Down syndrome, right? Because he had a daughter with it. And he always said, okay, when we get some good tools, maybe I will move to this field. And this was the case. So the first studies using environmental enrichment in the trisomic mice were done in Jesus' labs. And I had the pleasure of working with him at that time. So what happens with environmental enrichment? In fact, it works. So it works in transgenic. They get better learning curves. And what is interesting is that during this period, in fact, environmental enrichment also normalizes diagonal kinase activity. So if you look at the hippocampus of these enriched mice, you see that there is also a normalization of the diagonal kinase activity. Okay? We're kind of working through the same mechanism. Of course, there are many other mechanisms. I don't want to sound reductionistic. But, well, let's go through the whole thing, and I will explain you the rest. So we decided to see if we had synergic effects. And so there are 2 problems with environmental enrichment, as I said, like in humans. So the effects are less important than in euploid mice, first. And second, they last not so long, okay? So I cannot go through the whole thing, but basically what happens is 4 months after the treatment, the wild types are still having some effects. The trisomics do not have any more effects, and also the normalization of the diagonal kinase activity is fade away. It's disappeared, okay? So we try to do both treatments at the same time, and what we discover if we compare the enriched transgenic with the double treatment, what you see is that, in fact, we get better learning curves, okay? So we get better learning curves with the double treatment. It looks like they may have an additive or a synergic effect. We still don't know. And also, when we pair these 2 treatments and we look at the whole hippocampal region, you see that this phenotype that we saw is recovered in the transgenic mice. Okay, so this is fine, but we need to move to humans. And I don't know about the Eastern-Western differences about this, but probably we are more kind of direct in our approaches. And we decided to really go from this basic research to clinical trials. And basically because this was an extract, it's not considered a drug itself. It's considered a nutraceutical, so this facilitates a lot the whole approvals and everything. So we went into humans. And in fact, in this first study, what we saw is that it was safe. That was the first question, because things that are safe in normal population, in healthy volunteers may not be safe in diseased population. So we saw that it was safe. And in a phase 2 clinical trial, we decided to go for a combined treatment. So in the first treatment, we only administered epigallocatechingalate, 9 milligrams per kilogram. And in the second trial, we tackle also young adult population. In this case, the N was, in fact, we were aiming at 80 because the power analysis told us that this was a sufficient number to see the differences. But we had 87 in the end. And here, what we combined was EGCG with cognitive stimulation based in the studies in mice in which we saw that there was this additive effect between environmental enrichment and our drug. Okay? So this was a double-blind, placebo-controlled, etc. So everything normal. And here we had this design. So the first month was placebo for everybody. Why? Because we know that in this population, especially, the placebo effect is very strong. So people tend to see effects even though there are no effects, right? So we did a placebo during this first 1.5 months. And then we had 2 groups. So placebo or EGCG plus cognitive stimulation. So here the question was, is EGCG plus cognitive stimulation better than cognitive stimulation alone? This was the question, right? And we did the treatment for 1 year. And then we had 6 months of follow-up without treatment to see if there were some residual effects. What we did was we had biomarkers and body composition, psychology, and in a subgroup of patients, we had nose physiology and neuroimaging studies also. Okay? And well, this has been published in the Lancet Neurology this year, in May. And what did we see? In fact, we saw some changes in our battery. There were, especially in the adaptive behavior, functional academics, so the whole thing was significant, and especially in some of the tests. We saw also changes in the Down syndrome population. So one of the problems of the Down syndrome population, so Paul, we need your data somehow, is that there were no reference values, not for neuropsychology, because all these tests are developed for kind of other disorders, like Alzheimer's, for example, and they are not adapted for intellectual disability population. So this means that you have to adapt everything, and you have also to try to understand what are the baseline values for most of the things. So we saw deficits. Jesus Pujol, that is the person that did the study, saw differences in functional connectivity in different brain areas, basically in the SIB maps of the frontal, the Wernicke, and the cerebellum. And what he saw is that, in fact, the functional connectivity was improved after a treatment. And for example, this is one of the fMRI tasks. Here is these geometric figures that they also saw in their cognitive tests. And before the treatment, you see that basically what we see is activation of the more visual areas, and after the treatment, we recruit other more frontal areas. So in neurophysiology, we also wanted to have a more specific measurement of what was going on. And here, what we saw is that, in fact, well, this was a TMS study, so they received transcranial magnetic stimulation. And this can be used to measure the MEPS, of course, the motor evoked potentials, but it can also be used to measure kind of the cortical excitability. And this is done by doing stimulation and measuring at different intervals. So what the neurophysiologists saw was that, in fact, the distribution of intracortical stimulation and inhibition in Down syndrome, especially at the 15 milliseconds, was altered. And this was normalized by the treatment, okay? And this was not normalized within the group of placebo plus cognitive stimulation, cognitive training. So, I mean, this is great, of course, and we are very happy that it worked. But as normally happens, instead of closing questions, these kind of projects open more questions that they close, right? So one of our question was, okay, so why these changes in connectivity are affecting the functional properties? And this is very difficult to answer. But we made an attempt by using a graph-based generative model that help us then to calculate things like, well, sorry, here, I don't know. Oops, sorry. Here, like processing efficiency, storage capacity, and material metabolic cost. And this is the formalization simply in the equations. So here, what we do is we consider the neurons as nodes, and we can measure the connection, the connectivity. We can weight them, so we can make a directed connectivity, and we can put some weights in it, depending on if there are more or less, in our case, spines. That was the readout of the synapses. So it's a simplified model, right? It's simply to play a little bit with the parameters and see what happens. So we decided to take our data on the prefrontal cortex, and what we do here is we made an abstraction. So we use the dendritic tree size, and we calculate the synaptic contact probability, and then we also take into account the dendritic tree size variability, because this is something that we also have seen. And we parameterize our model with the real data that come from our experimental observation. So we finally end up with a graph of connectivity. Okay? The nice thing is that you can play. You can play with the dendritic tree size, with the synaptic contact variability, etc. Okay? And then this leads to what is called this efficiency space, in which you have the routing efficiency, the storage capacity, and the cost. And you can kind of put your points on of where your different groups are in the graph, okay? And based on all these different properties. So you can see here the wild type, that would be our kind of optimal system. And you can see how it goes a little bit further in routing efficiency and storage capacity after environmental enrichment. And then you can also compare where our Down syndrome mouse model is, right? So, and where does the environmental enrichment bring it to? So this is a kind of raw idea of what could be happening. Of course, it's just a generative model, so it cannot be taken as more than what it is, right? But here, what we saw is that, in fact, this trisomic layer has 25% lower storage capacity and 15% lower routing efficiency, whatever this could mean. Of course, when you look only at specific areas of the brain, and in many cases, at least in disease-related models, this has been done for many, many years. You constrain your observation to a very small, very specific area in the brain. I'm not sure if I will make it in 3 minutes. I will try. So what we wanted to have is a more general vision, okay? And of course, we could also do imaging, but it's not so good in mice, frankly speaking. We have done it, and it's... So we decided to go for this new system, that is this new brain clearing system, the Clarity based, because if you use the Clarity, every brain costs like 200 euros, which is not exactly in our budget. So we have improved our cost-efficient way of clearing the brains with TDA ethanol. And, well, you can have all these levels, right? A more macroscopic level, more population-based. And this is what we'd like to have, this population-based analysis. And if you want, I can explain you how we are going to analyze all this data, because this is tons of data. So we have already some nice images that we are starting to analyze to see this kind of mesoscopic range circuitry in these mice. So the second question, of course, is molecular question, right? okay, yeah, well, it inhibits Diagon-A, but how does it lead then to changes in plasticity? I mean, it could be through hundreds of pathways, right? And we did some proteomic studies. And what we see in the proteomic studies is that, in fact, there are pretty broad changes in protein abundance, also in phosphorylation, but in protein abundance also. And many of those are related to processes like plasticity, cytoskeleton, Rust signaling, translation, transport, degradation, etc. So the interesting thing, and if you want, later on I can show you more data on that, because we have also the phosphoproteomic data. But if you look at the profile of action of these different treatments, what you see is that, in fact, many of them are tackling proteins that are altered in the transgenic. But the one that is tackling more proteins, here in red, is the combined treatment. Okay? So from all these studies, we got some possible candidates, many of them associated with cytoskeleton. And here you see this is primary cultures of the brain. And here you see how the neurite of a wild-type mouse is traveling and how the neurite of this transgenic mouse is traveling. And in fact, what we see, oh, I get confused by this, is that the elongation and retraction movements are altered in these mice. And the responsible for that are the cytoskeletal proteins. So the cytoskeletal proteins are, in some of them are phosphorylated by Diagon-A, like tau, okay? And if we look at these cytoskeletal proteins, they are not only involved in the elongation of the neurite, but they are also involved in the changes in shape of the synaptic, of the dendritic spine, sorry. And here what we did is an experiment in which you have fluorescent actin. And then if you bleach this fluorescence with a laser, then you recover the fluorescence through the entrance in the dendritic spine of fluorescent actin coming from the dendritic shaft, okay? And what we see is that, in fact, the recovery of fluorescence is reduced in transgenic mice. And this gives rise to alterations in the dendritic spine shape. This is very important for the function of the spines. And the number of real contacts that we measure here using pre- and postsynaptic markers is decreased in transgenic mice, okay? Interestingly enough, again, when you use a Diagon-A inhibitor, you recover these phenotypes. So there are other questions. When? How much? Okay? I mean, you cannot think of Diagon-A as a single pathological condition, because it has different effects during development in the adult or in the elderly, because it's also related to Alzheimer's. Okay? So, and probably the most plastic time in life is the development. So we wanted to study all these things, but of course, this creates also a challenge. So how long should the treatment be? Should it be long life, or can we just give it through development and that's it, or what could we do? So to study all these things, and I'm almost there, okay. We need systems that allow these longitudinal studies. We need to understand the kind of natural history and how this is shifted by the treatment. And there are now some high-throughput phenotypic recording systems that are very useful. But of course, they pose a challenge to us, because we need to make sense of all this big data, right? And for example, we started with a very simple behavior, that is feeding behavior. And you know that in Down syndrome, there are obesity phenotypes. And one of the problems is their preference for chocolate and high-fat diet. They like that more than normal food, right? And well, this is, of course, this leads to increased energy consumed and to obesity phenotype. But of course, how does this start and how can we intervene? So we use this system in which they are submitted to high fat or to normal chow, okay? And this creates strings of very long data in which you can see, for example, the feeding bouts, okay, across time, how much they eat, and also the rhythmicity of these bouts. And how do we make sense of that? So what we have now done is we use genomic tools to plot our strings of data. And those are very interesting because you get a very nice visualization, and you can include here environmental elements like light and dark things and so on. But you can also zoom in or zoom out and see specific points in time. And this system is called Pergola, by the way. And you can, for example, use the circles. For example, here you see that in the beginning, all the mice do have this rhythmicity, this circadian rhythmicity. But once you put high fat in the trisomics, you see that they lose the circadian rhythm completely. Okay? And we can do many different analyses. I don't have time to go through all of them, but if you are interested, this is, of course, this is in GitHub. The code is available there, and it's open source for everyone that is interested in using it. And just to finalize, I just wanted to make a comment about the clinical trials, because you were mentioning the individual variation. And this is an extremely important problem in Down syndrome. Okay, and you have, at the genotypic variance level, you can have allelic differences, SNPs, epistasis. So we did some studies on that, profiting our clinical trial, right? And you see that there are differences in the cognitive and functional evolution may depend on many different factors that I don't have time to go through. So I just will mention that, for example, polymorphisms or variants in different genes, like dopamine-related genes that do have an effect on normal population, do also have an effect on cognition in our population. Or, for example, the levels of A-beta 42 are related with dementia-related markers. And we have found that semantic fluency is a very good measure for early prodromal dementia symptoms, or hypothyroidism that is also present in our patients. So you can imagine that all these, and finally sleep quality. So sleep quality is very much affected in Downs, and this also has an influence on cognition. So finally, we are confronted with all these different sources of variability that are affecting our population. And different to other populations, there are not so many patients here. And this happens not only in Down syndrome, but also in, for example, we have done the same study in Fragile X. We have very few patients there, right? So the problem is that this leads to differences in the effectiveness of the treatment. So in some patients, it's very effective, extremely effective, but in others, it's much less effective. And we need to understand why. So we need to really see why this therapy in different subpopulations has different effects, to go to somehow this kind of precision medicine, or let's call it like this. So the problem is the clinical trials as they are done now have many problems, are not effective, are not able to really sort out what is effective or not. So we need to really redefine what are the primary endpoints. And this is extremely difficult right now because of all the problems that had been in the past, right? But we think that we really need to redefine how we analyze and how we measure things, especially in intellectual disability. And we need also to analyze the factors which explain part of this phenotypic variability. And to this aim, we really need better biomarkers. So I'm finished. Just, I think that, well, I gave you some data supporting that plasticity-targeted treatments may be useful in these complex diseases leading to intellectual disability. But of course, this is not tackling a single abnormality. And we started by DRG1A, and we ended up by a proteomic study that is telling us that there are many more things involved, right? But probably some drugs are tackling these network hubs that are more important for the disease phenotypes. And what we think that EGCG is doing is not just gene doses normalization or gene activity normalization, but is really affecting other cascades that are also related to plasticity. And some of them are equivalent in other intellectual disability disorders. They are overlapping. And in fact, EGCG was also effective in a phase 1 clinical trial in Fragile X that we finalized this month. So, well, I will not go further on, but just to say that we may think in therapy not just through pharmacology. And I don't know how the pharmaceutical industry can include in their equation things like cognitive stimulation or cognitive training and things like transcranial direct current stimulation, for example. But I think that those should be considered in the whole equation. And just to finalize, because Balázs asked me to make it like more, well, broad and skip a little bit or go a little bit out of our niche, right? One thing that we should also ask ourselves as society is, what do we want to change and why, right? So we really want to be all the same, all normal, or what is normal, basically, no? This is a question that I can put to the audience. And one thing that we could also try to do is to think in terms of differences and not just of deficits, right? So, and I put this very raw example because it was in the last moment, but I'm always impressed by this, the case of, you know, this autistic child, Nadia, that had this savant syndrome. There are several autistic people that do have this savant syndrome. And she was able to draw these fantastic horses and horsemen at the age of 3. And if you compare it with a normal drawing of a child of 6, right, it's not exactly the same, right? And if you are interested, Lorna self-published a book on her. So, and if we go further with this kind of discussion, coffee discussion, we could think that many of the special people are those that are able to see things that the others don't see. And probably some of my colleagues here might be part of this collection of genius, no? Or different people. So I think that this is important. The other final message is that as a society, we also need to take into account our responsibilities. And we know that mental disorders are impacted by, for example, the economic crisis, the differences, the inequalities, the social inequalities, the pressure that we are putting on our families, on our kids, on our society in general, right? So probably we have to think a little back and see to what extent, I mean, of course, we will not change the economic crisis. I don't think that any of us, maybe there is some politician here, maybe, but the rest not. But at least we can change a little bit our surrounding world, right? Our little world. So this was just a reflection for well-being and the society. And just to finalize, I would like to thank my lab. This is Linus with the generative models, and Maria with histology, and Silvina with behavior, and then also, and I didn't mention the electrophysiology studies because, but I have the data if you like. They have reduced gamma oscillations. And this is the clinical team, and my colleagues, Mauricio Matia, Marcel Ruiz Mejías, Mavi Sánchez Vives, and Jordi García Ojalvo. And of course, we have to acknowledge also the funding sources and the anti-funding sources. This is the Ministry of Finances in Spain. So thank you very much. Thank you.
Thank you so much, Mara. So I think this is a new perspective here And I very much like your last conclusions regarding, do we really have to sort of squeeze the Gaussian distribution of our mental variability and psychological and cognitive variability in the population to a narrow bell curve, or
Yeah.
do we have to live with it or even accept it? So, questions from the audience, George.
Yes, bravo.
Thank you.
The thing is that you have made me travel in time back, because 12 years ago I was assigned to lead a lab team for my university, which was located in Shenzhen, which is across the border. And this was a lab that was under the supervision of the Ministry of Science and Technology of the People's Republic of China and of the university. So we had two saint patrons. And the lab function was to pick up natural substances from the traditional Chinese medicine, to analyze them thoroughly, to identify potential active substances, to make also whole extracts without supposedly active molecules, and to do a tremendous series of investigations in pharmacology in animal models. I mean, China is giving a lot of money for this kind of research. So our flagship, and there's a reason for that, in our department we have Bill T.H. Chan, who used to be the chairman of chemistry at McGill University. And Bill is probably the best specialist on EGCG.
Wow.
So we worked a lot. And he came up with an idea, which was to find the best product which would be soluble. That means you would absorb it, you would get it into your system, and it will go like bad girls everywhere. And it's an acetate. I mean, there's some trick in chemistry. It has been commercialized for the treatment, the topical treatment of skin cancer by a German company, and it works very well. It's a standard treatment now for skin cancer. The thing is that we tested all the commercial or commercially available EGCG, and first, some didn't contain any EGCG, which is quite common. Then, if you increase the dose of some commercial products, you get liver toxicity, because catechins are highly toxic. And if you drink too much very strong tea, when it comes out through the natural exit, it burns. So it tells you that it's not that good. And what I was first, Rafael de la Torre came to our lab 6 years ago with Maria Kovals. So we have a good connection with some people in your team. My question is for you and your team, if you were interested to pilfer our experience, and we can open all the books and everything, I think not only this product, not only this substance, but we have other substances which have the same type of mechanism in animals and in humans, and we know that. Except that because they are natural products, there is no money to be made by the by the pharmaceutical industry. So nobody wants to sell that.
Yeah.
And the companies that sell that are crooks. I mean, they want to make money also. They don't care about the quality of the product. So we have a concern, but we would like to show that substances with very good controls can be, what you said, saving lives and reduce the burden that the communities everywhere in the world have in a number of diseases, because all the physicians were trained with the money of pharmaceutical companies for the last 2 generations, everywhere in the world, in the Philippines, in many, many countries. So I think there's an opportunity to consider your pathway, which was ours, still is, and to see if we can do something better. Thank you.
Thank you very much. It's great news.
Excellent. Ruud?
Where the connection in the later slides between inflammation, obesity, high-fat diet, and worsening of the latency periods, for example, in these mice studies.
Mm-hmm.
Did you try anti-inflammatory drugs to see in this connection, so in mice that were fat and high-fat diet, because that generates a lot of problems in inflammation that is systemic and in brain?
Absolutely, and in fact, the main problem is that in Down syndrome there are some inflammatory genes that are altered also. So we are now studying all these pathways and how the inflammatory processes are. And diagonate is partially involved in those also. But there are many, many of the inflammatory processes that are altered. And they tend to have more inflammation and so on. Yeah, absolutely right.
Carlos?
I was wondering if you try to reduce the dosage of the DIRC1A in the mouse model, in the Down syndrome mouse model, not using the EGCG, but just reducing DIRC1A.
Yeah. In fact, I showed the data, but probably it was too fast. So we did some gene therapy. So we were using shRNAs against DIRC1A. And we were reducing it in the trisomic and in the transgenic. In the transgenic, of course, you only have overexpression of DIRC1A. So by normalizing the expression, even in the adult, we were able to normalize some of the phenotypes, mainly the cognitive phenotypes.
Also in the trisomic.
In the trisomic
okay,
also, and the LTP. So we were normalizing both LTP deficits and the cognitive deficits.
A penultimate question over there.
Yeah, I think we were all amazed by the drawing of the horses by the 3-year-old Down syndrome child. And it just got me thinking, is there any research that's looking at how we can enable talent like that rather than fix what is missing? And I know the problem with this, of course, is that mice don't draw. So there's going to be some, let's say, research issues here. But is there any research looking at that and looking to explain how is it that that can occur and what can be done then to develop that rather than trying to put back what we think should be there?
Mm-hmm. That's a very interesting question. In fact, with EGCG, one of the problems that you have is that it has also inhibitory properties on the kinase activity. And I'm now talking about DIRC1A, even though I know there is much more, simply because this is what we measured. So also in the wild types, you get a reduction. And the problem is that with these dose-dependent genes, when you go too low or too high, you have deleterious effects. So probably, if I would say what I would think that boosting natural plasticity, so really boosting the talent through non-pharmacological means, to me would be a better solution, let's say. Anyway, I think that your question has also this other component that I feel very important, And to me, and I tried to say this in the last slide, but this is something that is important also. So Down syndrome people do have alterations at the cognitive level, but instead the emotional system, so the social brain and their social behavior is really very good. I mean, they're very friendly, very empathic, and many of the parents are kind of afraid that we change this also, right? So if they get more intelligent, will they also get like more depressed or more stressed or whatever? So I think this is an interesting way of thinking about the problem. But I don't have the solution. What I can tell you is that by now what we have seen is that they get better in different domains, adaptive behavior, quality of life, that we also included in the equation. Because, you know, the problem with all these standalone tests, okay, you bring the guy to your office and you do some tests, and maybe one day he's not willing to do it, and they go, again, this test, come on, and so on, no? I mean, this happens all the time. And when you do repeated measures, you can really see this, no? So, in fact, I don't know, probably we need also a societal change, right? And integration, real integration, not this integration that we do like, oh yeah, I will get paid if I take a Down syndrome person or whatever. But instead really trying to understand what they can bring. And I don't know if you know this company, SAP, S-A-P. They do this like Oracle system for the finances or whatever. So these guys have the compromise of having 25% or something, not 20%, of their personnel with autism. And they have already more than 100 people with autism working for them. And apparently they are absolutely great in detecting code problems and errors. And I mean, SAP is one of the healthiest companies in the world. And there are many studies in economy showing that diversity in the personnel of a company really boost excellence and boost the economic healthiness of the company. So.
Mara, I would love to go on with these discussions because I think this is, I'm so sorry, but we really have to restrict our time because we have to start the next, I know you will kill me, but sorry for that. But really, I would say I cannot stand not to say one more thing. That I really think in this conference where we are also aiming not only at brain cognition but at good life research, the big questions you raised in the last slides and in the discussion are vitally important. What is good life in this context? Do we really have to boost cognitive facilities but probably at the expense of other human traits? And that's a question in schizophrenia also, do we have to reduce this sort of creative variability, which of course may result also with positive syndrome, negative syndrome, hallucinations and so on. But in that case we would lose Salvador Dali and name, I don't want to name how many other important creative people. So I think this discussion should have gone on, but sorry for the time limitations, we really have to start the next one. Okay. So thank you so much, but let's continue the discussions in the coffee break after the coming lecture. Thank you, Laura.